by targeting the components of Wnt signaling pathway. Brief introduction about microRNAs. These microRNAs are non-coding RNA regulatory molecules that are about 22 nucleotides long, and single microRNA, they can regulate thousands of genes and have also been seen involved in various biological processes like development, proliferation, and differentiation. These microRNAs, they get matured by two processing enzymes, which are DROSHA and dicer, resulting in the formation of microRNA duplex. Then out of these two strands, only one strand enters the RIS complex, which is RNA-induced silencing complex, and hence the target mRNA is either degraded or translationally silenced. In order to examine the regulatory role of microRNAs, we chose sea urchin as a model organism because it produces large number of gametes and we can externally fertilize them. And the embryos are transparent, so it makes phenotyping process easier. And in sea urchin, it has been seen that 49 microRNAs are annotated. That means that most of the microRNAs, they do not have multiple members within the same microRNA family, while as compared to the humans, there are hundred to thousands of microRNAs, and most of them have functional redundancies. By looking at the phylogenetic tree, we see that sea urchins belong to the phylum Echinodermata, which is a sister group of chordates to which humans belong. Moreover, sea urchin contains all the major genes that are present in humans. The life cycle of sea urchin begins when we externally fertilize them, and following different cleavages, we see 32 cell stage, then blastula, and then gastrula, where we see, at this stage, we see formation of three germ layers, which is endoderm, mesoderm, and the ectoderm, the outer layer. And then following gastrula, we see the larva stage, and then finally we get our beautiful sea urchins. Wnt beta-catenin signaling pathway is highly conserved pathway and has been shown to be important in endodermal specification as well. So in a typical Wnt signaling pathway, in the absence of Wnt ligand, this beta-catenin, it gets phosphorylated by the destruction complex, which promotes its ubiquitination and then targeting it for proteasomal degradation. While in the presence of Wnt ligand and when it binds to its frisile receptor, then dishevel gets activated and it inhibits this destruction complex so that there is no degradation of beta-catenin, but instead there is accumulation of beta-catenin into the nucleus, where it acts as a co-activator along with other transcription factors. It activates the Wnt responsive genes that are further involved in the formation of endoderm and mesoderm. And now in this area, in this particular area, the wedge area and the micromeres, the beta-catenin is present. So there is nuclear accumulation of beta-catenin into this region. So from here it starts off the endoderm and mesoderm specification process. So this stage is present in between it is a 60 cell stage which is present in between 32 cell stage and blastula. So in this embryo, in this particular stage, the upper half is the animal half and the lower half is the vegetal, where this animal half, it gives rise to ectoderm and the lower half, it gives rise to endoderm and mesoderm. Now specifically these wedge 1 and wedge 2 cells, these cells here, they give rise to endodermally derived gut, which is further divided into foregut, midgut, and hindgut, where foregut forms the esophagus, the stomach part, the mouth, and midgut forms the stomach and hindgut forms the intestine. In literature, it has been shown that beta-catenin is required for the endoderm and mesoderm specification. So when there is no nuclear beta-catenin, we do not see this endodermally, this is the vegetal view of the embryo, so we do not see endodermally derived gut, and we also do not see the mesodermal cells here, and only ectodermal tissue is left. But instead, when we overexpress beta-catenin, then we see excess of endoderm and mesoderm formation, but there is little ectodermal tissue left. So it shows that beta-catenin is required for endoderm and mesoderm specification. And previously in our lab, it has been shown that microRNAs are important for development, and global depletion of microRNAs have resulted in developmental defects. So when we knock down dicer, as we can see in panel A and B, there is reduced endoderm formation as compared to the control in dicer knockdown, and also there is reduced mesoderm formation. it shows that microRNAs are important. So we see similar effects when we knock down DROSHA as well, and that data is not shown here. So we saw that beta-catenin is required for endoderm and mesoderm specification, and microRNAs are also important. So we wanted to see this connection, this link. So this leads to my hypothesis that components of Wnt signaling pathway are regulated by microRNAs. So in order to test the microRNA regulation of beta-catenin, various reporter constructs were designed, into which the 3' UTR of beta-catenin was cloned downstream of renilla luciferase, and then later bioinformatically predicted microRNA binding sites were mutated using site-directed mutagenesis. And then once these constructs were ready, then the mRNAs were generated and they were microinjected into the single cell zygote. And we use firefly luciferase as a loading control. And the expected results would be when the microRNA they bind to the 3' UTR, there is a perfect sequence complementarity, then there is a basal luciferase signal. But when there is no microRNA binding site, because we mutated the 3' UTR, there is no microRNA binding, so we see stronger luciferase signal. So this is exactly what we saw when we mutated the first microRNA. This is in the 3' UTR, which is May 2007 at first position, or May 2007 at second position, or when we created the double mutant, or when we took the MADEEP2, the another microRNA, and when triple mutant was created, we see increase in luciferase signal as compared to the normalized to wild type. And similar effects were seen. So this is the graph on the right is the later time point. This was the 6-hour, this is the 15-hour post-fertilization. We see the similar effects, which is the increase in luciferase signal normalized to wild type. Importantly, the difference here in between these two is the May 2007 at second position, it seems to be more functional in the later time points as compared to the early time points here. So seeing these two microRNAs, the May 2007 and MADEEP2, in the phylogenetic tree, we see that the May 2007 is present in the echinodermata and hemichordata, and the MADEEP2 here is present in the sea urchins, and this, importantly, this has the same seed sequence as the May25 that is present in the human that regulate the beta-catenin in humans. So until now we studied the impact of microRNA regulation by creating artificial system, by creating the luciferase reporter constructs and then microinjecting the mRNAs. So now we wanted to see the impact in vivo, what impact it has in vivo. So for that, under normal conditions, we see that when microRNAs, they bind to the 3' UTR, it results in translational regression. And when there is no microRNA binding site, when we when we mask these microRNA binding sites by creating target protector morpholinos, so this results in failure of translational regression because microRNA cannot bind. So hence results in increase in protein. So using this assay, we saw that there is increase in beta-catenin level protein in microRNA TP treated embryos as compared with the control, which is a 2.5-fold increase. So this explains that when there is no microRNA binding, then the level of beta-catenin protein is increased. Now we wanted to see the blocking impact of blocking microRNA regulation of beta-catenin at molecular level. So for that, the qPCR analysis was done, and with the control morpholinos and target protector morpholinos. And here three different colors here represent the three different germ layers, which is endoderm and mesoderm and ectoderm. And these are the downstream genes of beta-catenin. And here each square represents one replication with 100 embryos. And this thick black bar line here shows the average. So we see here that the, like for example, the endodermal genes, the level is increased, they are upregulated. For example, the FOXA, there is more than 5-fold increase. So we see that the blocking microRNA regulation, it has impact at beta-catenin, it is upregulated, and also its downstream genes, they are also upregulated. Next, we wanted to see the impact of elimination of microiron regulation on endodermal